just so we know who's coming to these seminars. This is our final seminar series presentation for this year, and we saved the best to last.
Wow, that's a lot of responsibility.
So our speaker today is Jesus Fernandez, Bayo?
Yeah.
Who is an assistant researcher in the Biological and Agricultural Engineering Department. This is an appointment that he's received in just the last 2 years. His research has been focused on organic waste stabilization, environmental fate of pesticides, and the effect of amendments on pesticides and soil quality. Today he's going to be speaking about something that we're really excited about, because this was in response to a rapid response program that we have through the Western Center for Agricultural Health and Safety. And this was one that we particularly thought was good and funded, but we only gave him 4 months of funding. So it's remarkable the sorts of things that can be done with that kind of funding. And Heather, how much was the funding? Maybe
7,500?
7,000.
Yeah. So look at what can be done with just $7,500. So, but Jesus said that he would be happy to have people ask questions during his presentation, or if he would, or we can hold them off to the end. But I will go ahead and let Jesus start and introduce the topic of his talk today. Thank you.
Thank you. Hi everyone, thank you for coming. So this presentation is one of the goals of that small project. That is like to kind of spread the word of different alternatives to chemical soil fumigation. So the title is Introducing to Alternative Chemical Soil Fumigation, and I'm going to go through like why and what are the best ways to apply and how. And why we need to fumigate soils, or why we need to apply pests. We need to apply pest control because they are bad for our crops. And there is estimation out there that we can lose more than 50 or 60% of our crop production if we don't try to control this. Being one of the worst at the weeds, followed by insects, and some other diseases like nematodes, bacteria, fungi, etc. So we live in a continuous war. We need to fight this every day. And just for you to have an idea of the scale of the problem is in California in 2016, they applied 208 million pounds of pesticides. It's too large to like imagine.
Does that include herbicides? It's all the same thing?
That's include all the pesticides, or the general. So pesticide is the larger word for herbicide, insecticide.
Okay, thanks.
So just for you to have an idea of the scale, it's like equivalent to 7,000 heavy trucks just in 1 year. So it's a lot of things, a lot of compounds that are being released in the environment. And when we talk about pest control, one way of doing it that is targeting the pests that are in the soil is the chemical soil fumigation. And what this technique does is to apply compounds that are very volatile, so they need to keep it in the soil by covering with a plastic tarp. And they are quite efficient in controlling different pests. But at the same time, because the compounds are very volatile, the danger is different, because people who apply the pesticide, they need to have a lot of PPE, protective equipment, and regulation doesn't allow to use them in close to populated areas. So they are harder. They are environmentally, or from a health point of view, they are like kind of more dangerous, specifically because of this volatility. And when I was talking about war, one of the fungicides was used in the Second World War as a chemical weapon. So imagine how dangerous they are. but not only for the health, also for the environment. And one example is the methyl bromide. Methyl bromide luckily is banned, and it was banned because it was very dangerous to the ozone layer. And this is kind of giving some hope, because this is one of the examples of the methyl bromide where it's a global problem, and very different international countries, they can agree and make an agreement to ban a specific chemical. So knowing that we need a lot of international agreement, this can be an example of success and giving us some hope for the future. But yeah, agriculture is facing a lot of challenge because we are in a growing population. We mean we need supply, we need to get more food, and also the extensive agriculture is having negative impacts in the soil health. And also, it may not seem related to this talk, but we are generating a large amount of organic waste that we need to manage. So those are the challenges that agriculture is facing, and there are some alternatives to chemical fumigation that can help to minimize those problems. And so there are different alternatives to chemical fumigation. I'm going to talk about them today. One of the most basic is called soil solarization. Don't worry about the words, I will explain everything more in detail. And another alternative is known as anaerobic soil disinfestation, and then another that integrates somehow both of those alternatives is soil biosolarization. And they are all based in similar mechanisms as in fumigation, chemical fumigation, because they all utilize plastics. So let's now imagine we are a farmer in California, in the south of California. What happened there is that it's really hot in summer, really hot. So hot that farmers they can't crop anything there. So they need to keep bare soil, but because of the temperature and the high solar radiation. So these conditions are ideal for soil solarization. And what they have to do is to irrigate the soil and then cover it with a plastic tarp. And this plastic tarp has to be transparent, so we can have a greenhouse effect. So the solar radiation is going to go in, and then the wavelength is going to change, and it's going to be trapped as a greenhouse effect. So we can reach temperature at the surface that can be above 140 Fahrenheit. And that's going to be our main mechanism of getting rid of our pest. However, if we are in the coast, temperatures are milder, especially in summer, and so we can apply this high temperature in the soil. So what we apply here, the best way is to use anaerobic soil disinfestation, and in this case, what we add first is an organic amendment that is going to give us some kind of carbon source for microorganisms. And then we irrigate the soil again to activate the microorganism, the microbial community, but also to move oxygen from the soil. And we cover with a trap, a plastic tarp. In this case, it doesn't need to be transparent because we don't need the high temperature. It can be different colors depending on the brand, but the main goal is that this plastic has to keep the soil anaerobic. It has to prevent oxygen to go into the soil. So the mechanisms here are two. One, we have a significant decrease of oxygen. First, because we move it with the water, but second, because the microorganisms, when they start eating, they start breathing, they're going to take the oxygen, and they're going to consume the oxygen. And because there is no exchange, then we have these anaerobic conditions. But the other main process that is happening is that we are generating some kind of biopesticides. Because we have an anaerobic condition, we have some fermentation in the soil. This organic matter that we have added, the microorganisms are going to use it as a source of energy, and they're going to generate biopesticides. They are completely natural compounds. One of them is acetic acid. This is what we find in vinegar, for instance. Then we have propionic acid. Is that give us nice smell when we sweat. And butyric acid is another important biopesticide that is generated and it's like you can smell that, for instance, in butter. So those are the main mechanisms that are going to control our pests. But if we are now in the Central Valley, what happens is that we have hot summers, but actually it's the best season for the farmers to crop, so they don't want to keep the soil covered in plastic the whole summer. So we need to be more efficient, but we want to utilize that solar radiation. So what we do is to mix both techniques. So we add organic amendment, we irrigate the soil, and then we cover with a plastic tarp, having both processes. Because the plastic tarp is transparent, we have the greenhouse increasing the temperature, but because we have organic matter and we have microorganisms, we have this release of the biopesticide that are going to control the pest and hopefully decrease the time that we need to apply the treatment in the soil. So as a summary, which are the main factors that control these techniques? One is clear, is the weather, the climate. We need high solar radiation in some cases, and if we don't have them, we have to find an alternative, like anaerobic digestion. Time, the longer we keep the soil covered, the more efficient it's going to be. Depth, especially in solarization, where we have the hot temperature at the top, the lower layer is going to be harder to control the pest. Soil is a very important parameter. There are soils that are sandy, so they have more porous, so the porosity, so they can they are harder to keep anaerobic conditions in this type of soil. However, if you have a clay soil, then the water is going to be more stable in the soil, and then it's easier to keep the anaerobic conditions. But on the other side, after we have our treatment and we remove the plastic and we want to put a crop, we need to allow some reequilibration. So if we have a clay soil, it's going to be harder for the soil to lose all this organic acid that we have generated. But if we have a sandy soil, for the oxygen is easier for the oxygen to get into the soil, so it's going to be shorter the time that we need for recovering the soil. The plastic tarp, transparency, impermeability, a lot of different conditions that they're going to affect. An interesting one, you can see they can generate some kind of condensation in the water, and they may affect our efficacy. And last one, and the most important for our research, is the amendment. And the way we talk about amendment is the soil organic matter that is added to the soil, is the degree of stability. And I will explain a little bit more what I mean by that. So in general, we have a research objective that has been always to see which is the impact of different organic amendments in the efficacy of soil biosolarization. And we can do that at different scales. We can bring the field to our lab, and then we can use small incubator and small flasks where we can easily simulate biosolarization conditions in the lab that allows us to run a lot of different experiments to have a first overview of which one can be the best treatment. We can have an intermediate step, which is what we call Mesocosms. This is like 1 gallon size bags, so we can integrate our samples in the field, so we have the opportunity to run a lot of different samples, but we can play with original environmental conditions. And finally, the most challenging one is to run large field experiments. The size of this one is 900 feet every row, and they are more challenging because you really need to think well which organic waste you want to use, because you're gonna need a lot, and a lot of plastic. So you want to use something that you have tested in the lab that is going to work somehow, but then you're going to have all the heterogeneity of working in the real field conditions. The other thing that with chemical fumigation is that they are not specific for any target, or the alternatives is that we target every pest that is in the soil, which is good because then if we have weeds and we have nematodes, we can control both with one shot, let's say. And in our experiments, we have focused our research in nematodes, in fungi, and especially in weeds. Let's talk a little bit about the organic amendments and how I describe them according to the stability, the degree of stability, which is basically the biological activity that they have. The way we can measure the biological activity is by measuring the respiration rate. This is CCER, is the CO2 evolution rate. So the lowest is the CER, the lowest is the microbial activity, biological activity. So in the lower end, we're going to have organic amendments that they have been treated like compost or digestates. In the process, all the available organic matter has been already consumed by the microorganisms. But in the upper end, what we have is raw material like tomato biomass. So they have a lot of sugar as available carbon that they're going to be very easy accessible to the microorganisms. And we can play in the range between both extremes, because the one is going to be maybe too low, the efficacy is going to be too low, the other, the efficacy is going to be too high, but maybe we have some kind of other problems like phytotoxicity. So we can play in between both ranges by playing with the stability of the compost. So we take a compost process and we stop it before we get a mature compost, where you're going to have still some kind of available organic matter. But the other way of controlling that is to using lower amendment rates in the field. So taking into account these two parameters, we can play with the stability of the material that we want to apply in our experiments. And of course, in our lab, we have tested not all possible, but a lot of different ways. We have worked with compost, with digestate, with tomato biomass, almond hulls, and green waste, and some others. And they all behave somehow different. It's very interesting. So I'm going to show you some of our results, general overview of the results that we have. I have classified them in 3 different blocks: results regarding to the mechanism of soil biosolirization, results related to the efficacy of the process, and then another one important thing that we've been checking is the impact in the soil quality, in the soil properties. Regarding mechanisms, if you remember, we have, let's say, 3 main mechanisms: temperature, oxygen level, and release of organic acids. These results is a Mesocosms experiment where we have some temperature loggers in the Mesocosms, and this is the profile of the temperature evolution during the experiment. Of course, there is a significant cycle in temperature because of day and night. We can reach, this is at 5 centimeters. We can reach a temperature of up to 45 degrees Celsius. And when we look at 15 centimeters, we have a lower oscillation of the difference between the maximum and lower temperature are less significant. And the way these things change is going to be also affected by the type of soil. If we have a porous soil with air and not enough water, then the heat energy transfer is going to be different. But in this figure, what we see is the maximum air temperature, and that we prove is that our goal is to heat the soil at 15 centimeters. We have temperature in the soil that are up to 10 degrees higher than the maximum air temperature that was recorded in that day. So we can confirm that we have some kind of heating, which is what kind of expected. Regarding oxygen level, people usually measure redox in the soil. We've been able to set up a system where we could measure oxygen directly in the soil by sampling the air in our systems, and that's what is shown here. Is the oxygen concentration with time. Just remember that oxygen level in the atmosphere is 21 So a few hours after the treatment started, we have a sharp decrease in the oxygen level at both depths. And they kind of kept doing the experiment, but it seems that there is a recovery in the lower layer, and it seems our hypothesis is that there is some kind of diffusion of oxygen from the bottom of the soil to the top. Because in this in the top layer, there is no potential source of oxygen. So that's what explains why it's more stable in the top layer than in the lower layer. And then organic acid accumulation, we've been able to measure in HPLC these organic acids. This figure shows at around 5 centimeter the acetic acid, which is one of the most common organic acids that we find. We observe a peak in the amended soils. So this experiment was run with partially stabilized diastates and compost. So those are the three. And we observed some organic acid accumulation in those treated soils. And when we compare to the non-amended soil that was soilized, there is no BF phase. And this is at the top layer. At the lower layer, look at this. Differences are huge. There is a much higher accumulation of organic acids, and the peak seems to be reached after 8 days. And this is great because at the top layer we're going to have the heat that is going to kill the pest. But at the lower layer that we don't have that heat, we're going to have a higher accumulation of organic acid, and that's going to kind of balance the control of the pests. And let's see how this works, if it works. So, first of all, let's talk a bit about how it affects weeds. This is a figure of mortality, seed mortality of Brassica nigra. It's also known as black mustard. And in this experiment, we use digestate. So you remember we are in the lower end of the biological activity, more stable. And we don't see a huge impact. So these are samples that were incubated in room temperature to avoid the heat effect. And these are samples that were solarized. And the control are the black, non-amended. And the only place where we observe some kind of effect is in one of the solarized, one of the digestates. So not huge impact. If we move to samples that are more unstable, partially stabilized organic matter, then the trend is more clear here. So we have similar room temperature, not really affect. Solarized, we have an increase in all the samples that were amended compared to the sample that was not amended with the partially stabilized organic matter. And of course, the most unstable tomato biomass. So this is what we are seeing in this figure, is the mortality, but this is a kinetic study using tomato biomass. And after 5 days of using that amendment, we kill all the weeds. That's something that didn't happen in our samples that were incubated at room temperature, or the samples that were not amended. So this proved a high efficacy in controlling these kind of weeds. I went a little bit further. I wanted to know if I could some kind of final correlation between this stability degree and the mortality. So I gathered all the values of all the experiments, and I plot the respiration rate, that is a variable that we can measure in the lab, and the mortality, and there is a good correlation. And this is the samples that were incubated at room temperature. When we plot the same in samples that were biosolarized, this correlation is even better. And I went a little bit further, and I did the same plot with organic acid. And this is, I really like this, because this is a proof of how important, or how is the biopesticide activity that these compounds have. There's a very good relation between the organic acid level and the mortality. And these are the samples at room temperature, so we don't have the heat effect here. When we put the heat effect, because we are overlapping two different mechanisms, so the correlation is not as clear, but this is still a very good correlation. how this technique applies to fungi. So this experiment is again with the partially stabilized organic matter, and we had the chance of counting with a plot that was already artificially infested with a fungi, Fusarium oxysporum, which is in this case was specific for lettuce. So this is the CFU, the colony forming unit, which is basically the concentration of this pathogen in the soil. Initially, we see that our treatment were infested. When we treated the samples at room temperature, we don't have a significant effect. But when samples were solarized, we have a significant decrease. But this decrease is even higher when they were solarized and amended with the partially stabilized organic matter. And the same was observed at lower temperatures. So again, promising results on improving efficacy. Another thing that we have studied with nematodes. Nematodes are trickier because they move. Fungi and weeds, if it's hot, they can run away, but the nematodes they can't. So they are a little bit more tricky. And what I'm showing here are results of exposing the nematodes to the water extracts of soil that were incubated in conditions similar to biosolarization. So what we did was in the lab incubated a soil that was amended. In this case, it was amended with almond hulls. We incubated for 8 days. Then we did a water extraction, and then we put the nematodes in contact with the water at different strengths. And we observed that the nematodes rapidly died when they were in contact with the extract, confirming the nematicide effect of the extract. But when we diluted that extract, we didn't have that significant effect. And they helped us to decide which was the amendment rate that was used in this large field experiment that we ran. So as I said, these rows have 900 feet, and every row is like 10 feet wide. So we have different treatments. Some of them were amended with almond hulls. Some of them they were not amended, and some of them they were not even covered as a control. Some of the things we are doing a lot of things in that experimental plot, but one of the things we did was to measure the parasitic nematodes. In the controls that we after 10 days of treatment, in the controls that were not covered with tarp, we still have a high levels of nematodes, but in samples that were solarized or biosolarized, they almost disappear. But if we zoom a little bit, we still find at the lower layer we still find some nematodes that escape from our treatment. Something that didn't happen in our treatment with our most unstable organic matter. So again, promising results. And finally, the impact of these techniques in the soil health is very important, because one of the ways we want to promote these techniques is that because not only we are decreasing the addition of chemicals into the environment, but we are also somehow improving some of the properties of the soil. And some of the things that we have observed so far is that we can increase the field capacity. Field capacity is the capacity of the soil to hold water that is going to be available for the crops. So it's a very important property, especially in California when we have to fight for the water. So in samples that were amended, biosolarized and amended, we have a significant increase of this property compared to the non-amended. We also monitor the nutrients, the NPK, these nutrients they need to be available for the plants, and we observe that some of them they are increased after our treatment. One of them is the phosphorus, and this is again our control, and all the samples that were treated with biosolarization, they have a significant increase of the phosphorus level, and something similar was observed with the potassium level. This is very good. So farmers, they are going to need to apply less fertilizer to the plants. So sometimes people think that this technique can be too expensive because of the application of the organic waste. You need to amend the soil twice. You need to use the plastic. But if we add up all the benefits, then in the long term, it's going to be much better. And finally, the thing I like most is the microbial community and the soil microorganisms. People think that when we are doing this technique, we are sterilizing the soil. So the farmers, they are really concerned about that, and they don't like that. So what I did was to measure what is the microbial biomass in the soil after they were treated with biosterilization. And so that's what is shown in this figure. This is the microbial biomass in our samples. The black is the natural soil. We have a level of biomass. And when it was treated with solarization, not amendment, we have a slight negative effect that it was logically higher at the top layer. But when we looked at the samples that were amended, we have changed the scale. Look at that. We have a much higher microbial biomass. So we are adding a lot of microorganisms to the soil that are going to help in the cycle of a lot of nutrients. And we also have an impact in the microbial community structure. So this figure shows, it's like a map. It shows that if 2 points are closer, that means that the microbial community is similar. If they are far apart, then the microbial community are completely different. So this is our initial microbial community of the soil. When we add 2 different organic amendments, again, this is digestates, this organic matter is having a lot of different microorganisms. So obviously we are changing a lot the microbial community, and that's why they are so different. But what happens is that when we have the treatment, we are having a lot of extreme conditions in the soil. So only those microorganisms that are able to survive, they are going to stay in the soil. And what happens is that after biosterilization, we have some kind of merging of the microbial community, because only those that are able to survive those conditions are still in the soil. But they are still quite different to our initial microbial community. So, yeah, this is a very broad overview of all the experiments that we've done. And in summary, we can see that there is there are promising alternatives to soil fumigation, but they are especially dependent on the weather. So we need to know what conditions we have and what type of soil, etc., to know which is the best technique that we can apply. Particularly, soil biosterilization has shown to have better benefits than solarization, and that benefits are attributed to the degree of stability of the organic matter that we are adding, which is also related to the degree of organic acid that are released into the soil, in addition to other mechanisms that we have seen. Definitely, there are benefits in the soil quality that needs to be utilized, and soil biosterilization does not sterilize the soil. So what is left to do? There is a long way still, because we need a lot of data to know what is, for instance, the degree, the amount of organic matter that we want to have to, like when we apply a pesticide, we know the amendment application rate, so we need to have some kind of final amendment rate that we know is going to work, and there is a long way to have all that information. So for now, one of the things that we want to do is to improve efficacy at lower layers. As I said, nematodes, they can run away. So if we can move these organic acids through the soil profile, maybe we can follow and that nematodes, And so we are running some experiment in the lab where we have these columns where we have our amendment layer, and then with some irrigation practices, we can move through the profile the acids, and that's what we are doing, measuring the organic acid at different depths. And another thing that is important to have data is the potential phytotoxicity. After treating the soil, we have still this organic acid, and then they need some kind of recovery. And we are also adding some organic waste that may have or may not have some kind of phytotoxicity. So we need to do some more research in elucidating this kind of negative effects. Microbial resilience. So we've seen that we have changed the microbial community, but is that going to stay in the soil forever after we have treated? It's going to go back to the initial conditions, so we need more information about that. And also the long-term control. We have healthier soil, maybe crops are less vulnerable to pesticides, to our pests. So we need to apply these things with less frequency than usually. So just to wrap up, we are all looking for a sustainable agriculture, and I think what I like this culture, this type of practice, is that it helps that in several ways. One, we are reducing the pesticide application in the environment. It's helping us to add value to organic waste that other way may end up in landfill and generating greenhouse gases. We are keeping or at least improving or at least keeping the soil health, and that's why we need a more sustainable agriculture. And thanks. Of course, what I show is not only my work, it's the work of a huge group of people, a huge team that helped over the last years, and all this financial support. There's my contact information there, and thanks to this grant, we made a YouTube video that is out there, so you can check it if you want. And also, there's another YouTube video where we have our field experiment a little bit more developed. If you have other questions, and that's all.
Questions for Jesus? One question I have, when you put that plastic on, and it seems as though it only takes a few days for you to basically create the conditions that you want, then do you remove the plastic for then the growth of the crops, or does the plastic remain there?
That's a good question. It depends on the crops. So some of the crops, strawberries, strawberries mainly, they keep the plastic to have a long-term benefits. But usually the way we apply this is after our technique is, our treatment is finished, we remove the plastic, and then we allow this kind of reequilibration. We don't want to keep, for instance, in biosolidarization, we can keep a transparent plastic tarp because then we will burn the roots of our crops. So usually that's when in the strawberry fields what you see is a black tarp, because it seems that it's, you imagine that because it's black, it's going to be hotter, but it's not like that the case. Because the reason why we are heating the soil is because of the greenhouse effect. So we have black plastic tarp, we are just heating the tarp, but not heating the soil. And because of that, then in strawberries you can keep the plastic tarp and then keep your strawberries.
So just again along those lines, so once you do the application of the biosolarization, how long will the soil remain so that you don't have to use pesticides? Is this just for the season, or could next year, could we go, or do we have to do you have to place the plastic once again and go through the process?
So ideally that's going to, there are different factors that are going to define that, but ideally, as in traditional chemical fumigation, you want to apply this treatment once, and then you're going to use it after 2 or 3 years. So in theory, you shouldn't be using it every year. But again, it's going to depend on how rapidly our pest is able to recolonize. Pests are getting stronger, so sometimes that's changing with time.
So I was wondering, once you use this plastic, what do you do with it? Can you use it again, or?
Yeah, that's one of the people that things that people ask the most, because we are all against plastic now. So usually what they do is just they recover the plastic and they recycle it to produce more plastic. So it's not like that's the good practice. So the good farmers, they will take it and then will give it to the same company that lays the plastic can take them and then take it to a facility where they recycle. So that's what in theory should be done. But you can reuse it again, because because you can recycle but not reuse it. Because one of it gets torn with the heat, animals, and so you make holes. So it's very hard. And then the machinery that installed it is.
So I was also wondering, are there any emissions? I'm not sure if, like, for heating this plastic, do you have emissions of something you use either?
Emissions?
Yeah, like have you burned in something in the plastic that then maybe it's not significant, but.
You mean from the plastic? We haven't measured that. We have measured the organic, but you haven't measured from the plastic. No, we haven't. Can be interesting, but plastic seems still very, not very degraded at the end. So I'm hoping that it's not, but that could be something interesting to check.
You mentioned you were looking at the potassium and the phosphorus. Have you looked at all of the how it affects the nitrogen levels either in the soil or released from the soil?
We have measured only total nitrogen in the soil. We haven't measured emissions, which is another thing that has to be should be also monitored. In this case, we just measured total nitrogen in the soil, and we didn't see significant changes. Surprisingly, I guess because the microorganisms were utilizing, or because there were some emissions. So that's an important point to measure too.
Have you ever gone through the process of figuring out what the costs of biosolarization is versus just using pesticides?
So
we know that there clearly are health benefits, but what about the financial?
yeah, so actually there is a good paper out there where Chris Simmons made a life cycle assessment comparing biosolarization to chemical fumigation, and I don't remember which was the other. And in that study, they found that in terms of cost and greenhouse gases, it was more beneficial to use biosolarization than other techniques. But yeah, these techniques is going to be very dependent on the organic waste that we are applying, and if it is available and how far it is. So that's an important driver of the cost of this technique. And what we want to use is organic matter that doesn't have any value and is very close to our plots. So that's why we want to target as many different organic waste types as possible, so we can have different choices depending on the region.
I noticed you had several different, so I guess hulls and tomato. Is the one, I got the impression from you that tomatoes must be the most effective?
Tomato and almond hulls are the most effective because they have a lot of sugar and a lot of organic carbon that is easily degradable by the microorganism. So when we have this carbon, so the microorganism can take it and easily convert it to the organic acid that we are looking for. If we have green waste, where we have more lignocellulosic material, then this is going to be slower. We are having organic acid, but it's not going to be as extreme in that kind of waste. Yeah.
Sounds like strawberries are definitely on your list of.
Yeah, that's common, yeah. Next chapter.
Are there other crops? I mean, I thought almonds were part of this biosolarization almond orchards, or?
So the problem with orchards is that because they are trees, so the roots they go very deep in the soil. So and they stay there for long term. So we can. So this is more. This practice is more available for annual crops than for like long term, because even if we want to apply in an orchard that is still there, maybe we end up burning the roots or having some negative effects to the tree. So we can use it. That experiment was because we remove all the orchard, we went to plant a new one. So in that case it was okay, but still it's tricky because also the roots are very long and they go very down in the soil. So we are still not able to reach 5 foot of control, but we are working on that.
All right. So, if there are no other questions, Jesus would like to thank you for a very fascinating presentation today. And I'm sure he'll be here for a few minutes if you have additional questions. But let's give him a round of applause.